Okay, welcome back, biology. Today we're going to try a short lecture that looks at the characteristics of the cell membrane. I'm going to go over just some general things, why the cell membrane is important, so what it is, why it's important, and what it's made out of. And that's the part that is going to be a little bit technical, the composition of the cell membrane. So let's just look at a few general characteristics of the cell membrane. It is the barrier between the outside of the cell and the inside of the cell. And it takes us to a point that we've talked about before, and that is the importance of compartmentalization. So organelles are little compartments that store molecules of different types or create unusual or different chemical environments. We might create an environment in which the inside of an organelle has a low pH and another organelle might have a neutral pH, or we might store things in one organelle. Maybe we're going to store lipids in one organelle and store carbohydrates in another organelle. Well, the membrane is just one giant compartment that is storing all of the organelles as well as all of the resources that are in the cytoplasm. It separates the outside of the cell from the inside of the cell. The membrane is also a universal characteristic of life. All living things have a cell or plasma membrane, and so we can consider this a universal characteristic of life. And then finally, it is a selectively permeable structure. And what that means is it allows some molecules to move in and out, while restricting the passage of other molecules. And we'll talk more about how it's regulating things in another lecture. Today we're just trying to understand a little bit about the membrane in general. So let's just look at this nature of semipermeability and one of the main characteristics. I've set this membrane up in a way such that we have a high concentration of molecules outside of the cell and a low concentration of another molecule outside the cell here. I could alternatively have put a high concentration of molecules inside the cell compared to molecules outside of the cell. And just for the sake of argument, just because we have these red balls outside doesn't mean that there aren't any red ones on the inside. It turns out that the membrane is dynamic and flexible, but some things just can't pass through it very easily. So these molecules, which could be carbohydrates, they're just too big to pass through the membrane, and I've made them extra large to exaggerate it. Really, these carbs would be tiny, but still they are too big to pass through, and they might need a helper protein, some kind of protein channel that is embedded in the membrane that allows the carbohydrate to sneak through. But in the absence of these protein channels, we would say that the carbohydrate is not permeable and can't get through. On the other hand, these small molecules, and the small molecules that I've illustrated in red, they might be oxygen molecules. these small molecules are actually pretty small. And because they're so small, they'll be able to slide through the membrane. And the tendency is for molecules to travel from a high concentration to a low concentration. And we say that this is a downhill movement. So molecules cross the membrane from a high concentration until equilibrium is reached. And the process that allows this type of molecular movement is, pausing for you to think about it, diffusion. And if it is water that is diffusing across a membrane from a high concentration to a low concentration, then we call that osmosis. So when gases move across a membrane, gases like oxygen moving from outside the cell to inside the cell, following the concentration gradient from high to low, we call it diffusion. When it's water that follows the concentration gradient, maybe a high concentration of water inside the cell, diffusing out of the cell to a low concentration, we call that osmosis. So here you can see that the oxygen molecules have diffused and we have achieved some kind of dynamic equilibrium. We call it dynamic equilibrium because the oxygen doesn't stop moving just because it has equaled out. So these molecules might move around inside of the cell and they have an equal chance of traveling out as an oxygen molecule that is bouncing around, bumping into each other as another one moves in. So these molecules continue to be in motion, but they will diffuse until some kind of equilibrium is reached. So what is it about the cell membrane that allows these molecules to move in and out? Well, if we take this cell membrane and we bigify it, well, then we're going to have a structure that looks like this, and this is a lipid bilayer. And what this means is that the membrane consists of two layers of a specialized type of lipid called a phospholipid. And phospholipids are a universal characteristic of membranes. In fact, they are not only what make up the cell membrane, but they make up the membrane of the mitochondria. They make up the membrane of the endoplasmic reticulum. They make up the membrane of the Golgi body. They make up the double membrane of the nucleus, and so on. So if the organelle in a cell has a membrane, it's made up of phospholipids. So let's look at a little bit about what a phospholipid is. So I've got a phospholipid layer over here. But before I talk about phospholipids, I want to remind you about what lipids are in general. And so you can see I'm all set up to talk about that here. Lipids are large molecules. They're one of the big four. So the big four are nucleic acids, proteins, carbohydrates, and lipids. They're composed of carbon, hydrogen, and oxygen. Unlike water, they are non-polar. And because they are nonpolar, they do not mix with water. They separate from water. Lipids are attracted to lipids. So they love each other. But lipids are repelled by water. They don't like each other at all. And that's why water and oil do not mix. So these are large molecules, carbon, hydrogen, and oxygen, and they are nonpolar. Lipid characteristics, in addition to being nonpolar, they can be insulating. This is what blubber is composed of. They can be protective. You have a protective lipid layer surrounding your kidneys that protects your kidneys. Lipids are energy. And if you just think about the value of oil or gas in your car, you can see how this is energy in terms of fueling things, but it's also energy for your body. And then there's a whole bunch of different types of lipids. One type of lipid is referred to as a triglyceride. You might have heard of that. This is the type of lipid that people get concerned about when they are getting fat or they're accumulating fat in their body. A triglyceride has a small submolecule called a glycerol, which is a 3-carbon stem surrounded by hydrogens. And then coming off of that, there will be an oxygen, followed by a carbon with a group called a carbonyl attached, carboxylic acid, and then a bunch of carbons. And if you've ever heard of saturated fats, a saturated fat is a triglyceride with all of these carbons completely surrounded by hydrogens, and it is a big complex molecule. And if it is a saturated triglyceride, we're talking about things like butter and bacon fat. When these things accumulate in your tissues, they can clog arteries, create plaques, and give you heart attacks. They're not good for you at all. It takes a lot of working out physically to get rid of these, to burn these. There is a ton of energy in them. When people used to hunt whales, they hunted them for their blubber and used them to fuel lamps and heat their homes because triglycerides have so much energy. Some lipids are triglycerides, but they're unsaturated. Unsaturated fats are better for you. What it means to be unsaturated is that in that lipid stem, so these things called the fatty acids that are coming down, there are these occasional double bonds. And these double bonds make the lipids a little bit kinky. And when they get like this, what happens is the lipids don't form solids. Instead, they stay liquid. And when they stay liquid, they don't form into things like butter. Instead, these legs bump into each other. And we call these unsaturated fats. They're better for you. Do one more. And so this might be oil. I have not drawn a very good lipid here, but hopefully you get the idea. The structure is not quite as rigid as the ones that make up fats. And again, this is unsaturated. So we have saturated fats. We have unsaturated fats. We have waxes, oils, and the last group, which is an unusual looking group, the ones that look like this, and these are steroids. Steroids are lipids too, and all steroids have this basic structure. And the steroid that you are looking at here is cholesterol. You can tell it's a steroid by its name because it has that sterile stem, steroid. Testosterone. Testosterone. Again, you can see stero in there. Estrogen. These are all steroids. They all have the same basic structure that you're seeing here, and they can be important communication molecules. Interestingly, cholesterol is part of the cell membrane as well. And they keep membranes fluid, but if you have too much, they can make membranes rigid. And that's bad. This is good. Fluid membranes are good. Rigid membranes are bad. So let's look at the lipid of the day, which is the phospholipid. And let's see what's special about that. And we'll start that off today, and I'll finish this up with my next lecture. So when we look at the phospholipid, it's got two parts. It's got the fatty acid part. And these are like the legs of triglycerides. They are mostly carbon, hydrogen, and oxygen with some double bonds. That's why the legs are kinky. And the end result is they are non-polar. On the other hand, the top part, the part in purple, well, that's the part that we call the phosphate. And the phosphate is actually charged. And the end result is it's polar. So this likes water. And this hates water. We can refer to that in a different way. If it hates water, we say that it's hydrophobic. But if it likes water, we say that it is hydrophilic. So you might think, so what? Well, let's go back to the last picture where I showed you the membrane. And I'm going to erase all of these lines. Let's look at what's special about having this hydrophilic end and hydrophobic end. It's like I lost my legs with my phospholipids. Let me put those in really fast. Well, when we look at how a membrane sets itself up, it turns out that the surrounding environment of a cell is mostly water. And the interior of a cell is mostly water. So if you think about that, and we take a look at our our lipid bilayer, essentially what's happening is water is hanging out on this side, and no water is in the middle. So the end result is that the membrane naturally assembles itself in an aqueous environment. Again, just to color code a little piece of this, you could think of this black line as having, having these little purple heads, and purple heads on this side. And so long as there is water on the outside and the inside, well, we're all good because the hydrophilic heads are attracted to the water. And hydrophobic tails are going to be attracted to each other. And these membranes will self-assemble and stay stable. Well, that's it. This is my introduction to cell membranes. I want to encourage you to read and do your Pogles.